Welcome everyone to the Ali Houston Transforms podcast. I'm a former physicist who fixed my brain using nutrition, and I now coach people to empower them to do the same. And if you or anyone you know could benefit from a mental health tune-up, head over to metsci.com, where myself and psychiatrist Dr. Rachel Brown coach people to better mental health. And I'm really happy to have with me again Peter Dobromylskyj, who is a veterinary anaesthetist, surgeon, and nutrition blogger who created the legendary Hyperlipid blog. Welcome back on the podcast, Peter.
It's great to be back, Ali. Thanks for having me. That's great.
You're very welcome. Going back to our episode from 2019, it makes me think about going back even further to when I started reading about all this. And as a physicist who was unwell at the time, your blog really appealed to me because you were going back to first principles. I know that what part of what you're going to do today is kind of summarize this protons thread, which a lot of people love, myself included. And it makes me think about Dr. Myhill, who has a book called it's Mitochondria, not Hypochondria.
Yeah.
And how that comes up a lot in your blog, Hyperlipid, as well. Something I want to say at the top, which I did with the Brad Marshall podcast as well, and thinking about the feedback from our last episode, is that as with Hyperlipid, and any science, I guess, the devil is in the detail, but we will try to zoom out to recap whenever we can so that it's as accessible as possible without losing the actual meaning of the thing.
Yeah, fair enough.
So with that in mind, great to have you back on, and where would you like to start?
Shall we just begin with the cell signaling that as to how it wants to accept calories and how it should reject calories, and what it does with the calories once it's either accepted or rejected them. And that's Perfect. The core to where the protons thread came from. And as a kind of general principle, I have a concept that fatty acids, when you oxidize them, generate superoxide. And it doesn't matter really what state the mitochondria are in. Beta oxidation of fatty acids and feeding them into the electron transport, feeding their products into the electron transport chain, generates superoxide, which obviously is normally converted, if it goes any distance, into hydrogen peroxide. But it generates reactive oxygen species, and it does it roughly in proportion to how much of the substrate is coming from fatty acids. So fatty acids generate superoxide. That's one core bit.
Mm-hmm. Yeah, and just sorry to interrupt, but
yeah,
if this episode goes out after the Brad Marshall one, which I think it will, people should be familiar with this kind of cast of characters. If you don't have a scientific background and all these names are kind of make you scared and want to stop listening, please persist. They're really just names for things which you can understand the effects. You don't need to have that background to understand the flow of what we're talking about here. So can you say that again, that the production of superoxide is proportional to your fatty acid intake? Is that what you're saying?
Your fatty acid oxidation. You have to burn it. If you intake it and store it, it's not going to produce anything. But if you burn it, or if you burn the fatty acids, they're going to generate superoxide.
Yep.
Which is a signal. It's a signal that you're burning fat. If you run your metabolism instead on glucose, it generates far less superoxide, excepting when you take the products of glycolysis and feed them into the mitochondria from the outside through the glycerophosphate shuttle. That, I won't go into the hows and whys, but we can accept that makes glucose behave more like a fatty acid. Yeah, so it will then make the glucose, which generates very little superoxide, generate more superoxide. So we have a balance between fats being oxidized, which always produce superoxide, and glucose being metabolized, which under certain circumstances will generate superoxide. Okay.
So, and this is in the sort of wider setting of thinking
of everything.
The hyperlipid blog being, you can fuel yourself with glucose, you can fuel yourself with fat, at any mitochondria at any one time. They
will balance it.
Will balance it.
Will balance it. And the other very important thing that I carry in my head is that I'm not very interested in the brain. The brain is a kind of add-on to basic metabolism. The brain doesn't do anything different other than to basic metabolism, other than fine-tune it. Yeah, I can't see the brain being given a supply of calories and doing something different with them to what the mitochondria are going to do with them. So in the brainstem, as far as I'm concerned, there's a model. The brain has built a model that it feeds the energy supply that it senses in the arterial blood supply into this model and sees what it does. And it takes the reactive oxygen species, the superoxide that is generated in that model, as the satiety signal. Yeah, so the brain is mimicking what happens in the rest of the body based on what it's being given from the rest of the body. And it decides whether it needs more calories, less calories, or to waste calories, depending on what signal it generates in the brainstem. I wouldn't like to comment where it is, but people start with anorexic hormone neurotransmitters and orexic neurotransmitters. All of them, from my point of view, will be controlled by superoxide generation in the brainstem, in individual cells.
And this is something that is really important about the production of superoxide in terms of feeling full, the satiety signal. There's obviously like multiple feedback loops in terms of satiety, but this superoxide signal is system wide. So yes, it's your mitochondria across the body will be putting out this signal essentially.
Yes. But the brain sees it from its model that it mimics what's going on. There are nerve supplies to every cell in the body. All the adipocytes will have a nerve supply, so that you can have a neurological input. But the overriding one, as far as I'm concerned, is the model in the brainstem that uses whatever's being supplied in terms of calories to either generate or not generate superoxide. If it generates superoxide, it says stop eating. If it doesn't generate superoxide, it says keep eating. That's how I view the kind of superficial role of the brain. Yeah, but you can't. If you have a car and you put diesel into it, you could, and it should have had petrol put into it, you could probably tweak the engine control computer to maybe make it run if you tried very hard, but it would never run properly. Yeah. So the computer does what it can, but if you mess up the fuel supply, there's a limit to how much it can compensate. So
can you explain how this model is different from, say, another form of detection?
How do you mean?
So you're saying the brain has a model in place?
Yes.
Can you go into that a bit more?
Oh, well, this is just what I think. Okay.
Yeah.
If you want to know, if the brain wants to decide if there are adequate calories available, it has to, in some way, look at how much glucose it's being supplied with, how much free fatty acids it's being supplied with, potentially how much protein it's being supplied with. That's a kind of slightly controversial thing. And it has to try and integrate the whole lot together to see whether it's got adequate calories to do what it needs to do. There are a group of cells in the brainstem, which have lots and lots of glucose receptors, transporters, and it's got actively expressed CD36, so it transports fatty acids as well. And it can simply take the arterial blood supply, feed it to this group of cells, and see what they say in terms of superoxide generation. So if they've not got enough calories, they won't generate an awful lot of superoxide. If they've got tons and tons and tons of calories, they'll generate lots of superoxide, and that's then transmitted to a neural signal, depending on what the level of production is. And in fact, there are papers around where if you infuse intra-arterial antioxidants in the carotid artery to the brain, you can actually, in rodents obviously, you can suppress the satiety signal, and animals having their brains infused with antioxidants will just keep eating because they never get a satiety signal. And these things have driven me to think in these terms of what stops you eating.
And that brings us back round to glucose versus fat and the production of superoxide.
Yeah, it does. Because obviously you can produce superoxide from glucose, because even if you eat a bowl of white rice and another bowl of white rice, another bowl, you're going to stop eating at some point. The agricultural humans have eaten carbohydrate-based diets for thousands of years, and obesity is much more of a modern problem than that. So if you have the normal processing of glucose, correct processing of glucose, I don't see that anything needs to go wrong with it. I think if you stand back and take the much wider picture, running a metabolism on glucose and insulin clearly works perfectly well, doesn't make you obese, may not be ideal on a longevity basis, but that's a whole different can of worms. But essentially, it's not going to lead to metabolic syndrome. So
maybe deficiency if you're just running on rice.
Well, yeah, but ignoring the micronutrient basis,
all things being equal.
Yeah, you don't become obese.
Source you're talking about.
Yeah, and you don't become obese simply, from my point of view, again, by eating carbohydrate. There has to be another component to it, and we then come back to this whole business of the L6 polyunsaturated fatty acids, the fact that they do not generate as much superoxide as a saturated fat would, and that again came as a realization I had very early on in the protons thread. It came from Dave Speyer in I think he's in the Netherlands. I forget which university now, but he again, he's a deep thinker whose primary interest is in the origins of the eukaryote cell, and he loves peroxisomes because peroxisomes is where you put the fatty acids that make too much superoxide. It is possible to make too much superoxide, and his comment or his concept is that at the time of the formation of the eukaryote cell, bacterial fatty acids that were above 22 carbon chains long were automatically shunted away to the peroxisomes, where they could be oxidized without doing damage to the mitochondrial signaling. So there are a whole load of fully saturated, oddly named fatty acids that are peroxisomal targeted. Anything C22 and upwards is targeted that way. My first insight following on from that is that the fatty acids with lots of double bonds, this is DHA and EPA, and there are even longer ones, but they're
so-called good omega-3 ones.
Yeah, good in inverted commas. They're all shunted to peroxisomes as well. And of course, they're not being shunted to peroxisomes because they make too many superoxide molecules, because they're full of double bonds, which cuts down the amount of superoxide that's produced during beta oxidation. They're shunted because they probably don't produce enough, and they would mess up the signaling. So I have this concept that the very long ones are shunted into the peroxisomes. They're not really dealt with by the mitochondria. DHA maybe a little bit, EPA probably a little bit more, but the mitochondria are really dealing with stearic acid, oleic acid, and the C16s and the C16 monounsaturates. Obviously, they can deal with the medium chain triglycerides as well, and they do. But as a somewhat different set of problems I have is why medium chain triglycerides are very good at producing obesity in rodent models. And the question then comes down to why humans, whether they're obesogenic in humans or not obesogenic in humans, it's hard to call it, but I suspect that if people are heading into weight stalls, I would suggest that with low carbing, then you should, people say consider cheese, dairy, which obviously is full of medium chain triglycerides. And I would also be concerned about coconut oil, because coconut oil is one of the best obesogenic models in rodent models. Both coconut oil and butter, they're both, provided you add a little fructose, which I won't go into fructose, but I'm really not going to go into fructose. It's too complicated and I don't understand it. But they are potentially obesogenic models, and I think if you're having problems with weight loss, then as well as taking out L6s, then maybe taking out medium chain triglycerides might be worth considering. Ali, how did we get here? Because I've gone rambling off.
No, it's great. We're talking about eukaryotic cells, these early cells from when the complexity of life really on Earth went from bacteria to something more complicated. Yeah, we're talking a few billion years ago, right?
Eukaryote, 2 billion years, yeah, about halfway through evolution, basically. Yeah.
Sometime after the boring billion.
Yes.
And you start thinking about how long these evolutionary kind of the evolution of evolution. It's really hard to imagine those times. I think it's safe to say that several billion years is enough to try almost everything that it could have in that environment.
Yes.
And that the fact that these complex eukaryotic cells seem to have sprung up once because they share hallmarks that indicate that it only happened one time, or that the ones that survived came from a single happening. And this sort of brings us around a little bit to not only the fact that it means that if you can find effects from things like mitochondria, which are in these eukaryotic cells, which is really a sort of a bacterium that got into another cell, and the cell said, right, okay, you can live here as long as you produce the power for me.
Yeah.
Then you can take mouse models, you can take this well-developed theory of how mitochondria work and say this should apply to humans because we share these ancient mitochondrial processes up to a point. And thinking about Nick Lane and his writing on that sort of stuff, maybe that sort of brings us back to the protons thread.
Yeah, and ultimately, Nick Lane was hugely influential on how I viewed it, and obviously nobody ever agrees 100% with anybody. But he's and more even recently, has affected the way I think about things a lot. But again, we're talking about putting a bacterium inside an archaea or inside another bacterium. But the bacteria have been talking to themselves using superoxide. And it's very likely that the archaea have been signaling to themselves using superoxide as well. And all of the eukaryote adaptations have still preserved that signaling system. It was only quite recently I realized that bacteria still use superoxide as a primary reproductive signal. If you grow deep ocean bacteria, they will produce superoxide, and they'll grow given food. If you scavenge that superoxide, you can give them as much food as you like. They won't grow and they won't reproduce. And the superoxide is the signal. And that signal, I think, is still being handed from the mitochondria to the nucleus of eukaryote cell, or possibly into the cell surface. But these are very, very old signaling systems that may, I expect they go back to Luca, the last universal common ancestor, who, as far as we can tell, she was trapped in hydrothermal vents and wasn't free living, and only and escape from the vents to free living occurred on 2 separate occasions, once to give bacteria and once to give archaea. And because they're so similar, Nick Lane feels, and I see no reason to disagree, that they probably came from the same vent. And one end of the vent had bacteria and the other end of the vent had archaea, and they both developed techniques for getting away from the vent, which is how, but they are so similar.
By the way, I should say, for people who don't understand what a hydrothermal vent is, it's a hot vent under the sea, where there's perfect conditions for creation of life. If you're looking for the best possible place that it could have happened on Earth,
they're in disequilibrium. There is free energy available, and when metabolism started, it was almost inevitable. I mean, people think it probably started within less than 500 million years of the Earth being cool enough to have water on the surface, which is also interesting because that says when they start drilling on Mars, they're going to find bacteria-like substances below the surface because there was surface water. Once you've developed life, and the metabolism that is generated in the vents is likely to lead to life, it seems to happen quite quickly. Getting eukaryotes is a different matter, but getting bacteria or bacteria-like organisms, you'll have to drill down because the surface is very inhospitable. But a mile below the surface of Earth, there's a huge biomass of bacteria, and they're basically mostly living on radiolysis, where uranium decays and splits water to hydrogen and oxygen, and the bacteria a mile below the surface of the Earth will then recombine the oxygen and the hydrogen to extract the energy that was provided by the radiolysis. And they're there now. People are getting them off the tip of drills and investigating them. Fascinating subject. It's so yeah. I think that there is almost certainly going to be life on Mars, but you'll have to drill for it because once it's started, I think it's very hard to kill. But it won't. There's no environmental reason for it to be on the surface. It's going to do far better below. Okay, so I shut up about that now. How do we get to that one as well? Oh, right. I wanted to go on at some point. Let's just mention that the whole reason I hate linoleic acid is that,
which is one of these omega-6 fats that's highly prevalent in the modern food environment.
And they don't generate the superoxide as well as saturated fatty acids do. So the satiety signal comes later, and your adipocytes are a bit bigger before they decide to say, I've had enough.
Yeah, fat cells grow, that you can eat more until you feel full.
Yes, yeah. And again, they will be not only are they delayed in your fat cells, not only delay satiety in your cells, of your fat cells, but they will have the same effect in the brain stem. And again, you will then, you can talk about that as they're non-satiety generating because they don't generate superoxide as well, and that's protons ideas, and I think Brad has taken those ideas and really gone upwards, whereas I took those ideas and went downwards in terms of direction of, I'm more and more interested in the most basic processes I can find. Brad seems to have hacks, and that's great. That's much more practical than anything that I'll ever come up with. And it's a good idea for people to do these things. That's great. But there is a general anti-L6 feeling, and people come to these conclusions from different directions. I don't care. I have my own ideas of why I don't like L6s. If other people have other ideas about why L6s are bad, that's great. As long as we stop eating the damn things, that's perfectly okay by me. The more you look at the way other people look at the same problem, I can see the convergence, and I can see, well, I think this happens this way, and that would show as so and so. And I think ultimately the problem is that they probably are bad, and you can converge from several different directions as to why they're bad. I have my own ideas. And actually, I have to say that I don't listen to an awful lot of blogs or podcasts on this type of subject, because when I do, I just find I'm agreeing with people, and I really need to, A, I need to go and do my own thinking, because that's what I've always done. And B, you only really get interesting ideas when you disagree with somebody. That's when the progress comes. So that's, I went through a phase of listening to Lex Fridman quite a lot, but mostly on well, he doesn't really do metabolic podcasts, so apart from Nick Lane. So yeah, that was that. My feeling is that L6s, they're bad, and they lead to obesity as a consequence of their poor ability to generate superoxide. Yeah. The definition of generating obesity is that you have failed to limit the ingress of calories into fat cells. And as a general principle, that probably applies to all cells. And you then have to ask yourself, well, if there are too many calories coming into a given cell, people who are obese should be full of energy, should be spontaneously active, should be running marathons because they've got too many calories coming to their cells. And that to me has been at the back of my mind. That's been a very interesting problem. Particularly with the huge interest that there is now in metabolic psychiatry, Chris Palmer and yourself both talking a lot about it, made me re-examine the concept of why people with a predisposition to obesity should have poor energy generation. Once you become obese, as your fat cells get bigger and bigger and bigger, the ability of insulin to suppress fatty acid release goes down and down and down. And that's another whole story, but I won't go into that. But ultimately, we can accept that if your fat cells achieve a given diameter, the fat droplet starts to undergo lipolysis. It's called basal lipolysis as opposed to ordinary lipolysis, and it's not suppressible by insulin. It's augmentable by adrenaline-like substances. So clenbuterol, things like that. You can force basal lipolysis using clenbuterol and become very thin and well-muscled. But basal lipolysis goes up with the diameter of the fat cell, and it's not linear. It goes up on a kind of vaguely linear basis, and there's a certain size of individual fat cell where basal lipolysis suddenly goes through the roof. So you get to a size where of being overweight where your adipocytes just leak. It's not an accidental leak or anything like that. It's very actively controlled, but it just leaks. They just leak fatty acids. So, and that's not controllable by insulin. So you eat a high carbohydrate meal, your blood sugar level goes through the roof, your insulin level goes through the roof, and your free fatty acids should go through the floor. should go under 50 micrograms per 50 micromolar in the bloodstream, but they can still end up saying up at 1,000, which is fasting levels. So you've then got postprandial glucose, where there's a ton of glucose there. You've got insulin trying to facilitate you to use the glucose, and you've got fatty acids that shouldn't be there. So you're then in a situation, and different adipocytes will increase their basal lipolysis at different sizes. There's a spectrum, a range, or a normal distribution or probability, and there are a few chemicals that will affect it as well. So alcohol causes lipolysis that mimics basal lipolysis. Fructose, if you can get it as far as the adipocytes, will increase basal lipolysis. Some of the trans fatty acids, the artificial trans fatty acids, not so much the dairy bacterial derived ones, but the trans-industrial fatty acids, they'll all increase lipolysis. So you have these cells which have got a ton of glucose available, a ton of insulin available, and a ton of fatty acids available, and they should never be in that state. They should, if you've got high glucose, there should be low fatty acids. That's how the adipocyte is set up to control matters.
So this is a classic kind of modern obesity, broken metabolism situation.
You can't be flexible. You can't. What you should be flexible. You should either be burning glucose with insulin and shutting down fatty acid release, or you should be burning fatty acids because there is no insulin or very little insulin because glucose is low. And you should be able to flick between those two states, between fasting and eaten and postprandial. And if your adipocytes are too big, whatever too big means, it's not the same in every person, or at the same time, actually different. If you eat different substances, you can alter the response.
Mm-hmm.
Yeah. If you have too much, then ultimately you are going to supply too many calories to all cells in the body, and you should, once you're obese enough, stop being hungry. And that clearly doesn't happen. But theoretically, that's what should happen. So I had to go and revisit. I've had this at the back of my mind as a worry for a decade. Why do people who are obese, why are they still hungry when their fasting free fatty acids are very high, and their fed free fatty acids with glucose are still very high?
This abundance of available energy, it leaves them still hungry. And on the other side of the energy in, energy out equation, they're not feeling this spontaneous energy level increase or increase in warmth. So what's happening?
Well, this again, I go back to Nick Lane. He has been very influential on me, and he floated the concept that what cells care about is the flux, the rate of production of reactive oxygen species. And again, if you're somebody like me who thinks everything goes back to reactive oxygen species, and you go right back to the fact that bacteria are still using reactive oxygen species as their primary signal, and have been doing for 4 and a bit billion years, reactive ROS are the control substance, and they're what the body will and will not tolerate. They're what they use as a signal for everything from feed, grow, die. Yeah, that's everything from apoptosis is reactive oxygen species derived.
That's when a cell kind of they commit suicide, really.
Yes, but very technical, very controlled.
Mm-hmm.
Very much, this is exactly what we want to do. There are other techniques for dying, but apoptosis is the one which is nicely reactive. In fact, they're probably all techniques are probably reactive oxygen species derived. Apoptosis is a nice one. So if the body can deal with a certain amount of ROS generation. It's got superoxide dismutase, it's got catalase, both of which are enzymes that break down, converts superoxide to hydrogen peroxide, and then hydrogen peroxide to oxygen and water. So the cell has ways of dealing with it, but there are limits to what can be done. And the body would rather shut down ATP production if by shutting down ATP production, it can limit excessive reactive oxygen species generation. And
where ATP production is the universal unit of energy that the body uses.
Primarily produced by the mitochondria, produced by transferring electrons in as opposed to out, and then using the proton gradient of the mitochondria as like a water mill to produce ATP as the protons flow back into the cell. But the body will tolerate a lack of ATP if it has to suppress reactive oxygen species levels to a tolerable limit. And the research Nick Lane's group have done, not published yet, but it will be, I'm sure, is that they looked at the glutathiolation of complex 1. Glutathione, as we all know, is an antioxidant, and it particularly is joined if you have changes to the sulfhydryl group, the sulfur group, either on a protein or on glutathione, the two will join together, and you can stick glutathione onto a protein if that protein has been damaged by a reactive oxygen species molecule. And if you have too many reactive oxygen species being produced by the electron transport chain, the body will add glutathione to the complexes, the mitochondrial complexes, and simply shut them down. And it particularly shuts down complex 1, which is complex 1 is the one that uses NADH, which again we don't have to worry too much about, but NADH is a universal currency, and it's the one primarily produced by glucose. Glucose oxidation. Glucose-derived calories tend to produce NADH. Fatty acid ones more mixture of NADH and FADH too, which is another energy currency. Again,
like a sort of electron carrier. You know, you think about,
yes,
you mentioned the water mill analogy. If you look at these things, they actually look like little machines, little rotating machines, little machines that push the electrons through. They do, yes. There's these substances like NADH and so on that carry the electrons, and they're specific to different processes, like you say, whether it's glucose or fat that you're getting the electron from. But all people who don't have the depth of background have to worry about is there's little machines in your mitochondria that strip the electron and move it along, and that these different NADH, FADH electron carriers are there in different proportions depending on which type of fuel you're using or trying to use.
Yes, but if you are providing simply too much fuel, i.e. you've got glucose at 20 millimoles per liter, high fasting postprandial glucose, and you've got fatty acids at high concentrations, and you've got insulin facilitating the generation of reactive oxygen species as well, you've got too much. And if you've got too much, the body doesn't care about getting tons and tons of ATP at that point. It just wants less reactive oxygen species to be generated. And if the easiest way to do that is simply to shut down complex 1. And it glutathiolates complex 1. Complex 1 then stops transmitting electrons down the electron transport chain. The voltage component, delta psi component of the mitochondria goes down, and reactive oxygen species generation is limited. And the body appears to be perfectly willing to mangle ATP production to limit reactive oxygen species generation. So you then have an electron transport chain where the front end of it is basically turned off, and the rest of it is still working. But obviously the complex 1 produces a huge proportion of the membrane voltage in mitochondria, and simply disabling it will drop the membrane voltage, and it's very hard to generate reactive oxygen species if there's a low membrane voltage. So
And if you can't generate reactive oxygen species, it's hard to feel full, right?
Yes, yeah, absolutely. If you have this happening in peripheral cells, we call it insulin resistance, because you've shut down reactive oxygen species to avoid going over the top. In the brain, you would simply describe it as hunger, yeah, because the satiety signal doesn't come. And when you start with, when you go from being obese without insulin resistance, i.e. you're not leaking too many fatty acids from your adipocytes, to obesity with insulin resistance, then the types of fatty acids that are being produced are whatever is stored in the adipocyte. It's not selective. It's not influenced nearly as much by diet and the meal you just ate a moment ago. And if you've been eating a high carbohydrate diet and your fat cells are full of palmitic acid with some oleic acid, that's what will get released. So these are really good, particularly palmitic acid and stearic acid. They're really good at producing reactive oxygen species. They will be being released in combination with glucose. The cells that are getting this delivered to them just can't cope with it. They don't want that many reactive oxygen species, and they shut down complex 1. They also do interesting things to complex 4 as well. But again, the general principle is cells are willing to shut down ATP production to avoid excessive mitochondrial reactive oxygen species being generated.
So it's a protective mechanism that comes at the sort of this endpoint, obesity, of some process or set of processes that lead to obesity, and it's a kind of ironic checkmate. If people are eating like that,
yes,
then their fat cells are leaking the very type of fat that is making them that should make them not hungry and
yes,
and give them a source of fuel, but something's not working.
Complex 1 is being shut down because they would be producing, there are too many calories, and it's impossible to limit the calories at the adipocyte end of the supply. So the evolution has decided that simply shutting down ATP production is better than having too much reactive oxygen species generation. And hence you simply don't have energy. And that classically is a problem that people who are obese struggle hard. Do you remember from, might have been before your time, there was a blogger called Jay Stanton.
The name rings a bell.
Yeah. Gnolls.org, and he signed off. He's a low carber. He said, I've said all I need to say. I'm burned out. I'm going to stop blogging from now onwards. And that was 12 years ago, 13 years ago, 12 years ago. He was a nice chap. And we talk about reward and addiction and things like that. And he suggested that if you were sat down and fed a dehydrated soybean meal, chili con carne, whatever that was you'd been backpacking around, if you were to sit down and eat it now, it would be disgusting. He said if you had a long day in the hills, you'd covered a lot of miles, you were ravenous, this would taste like ambrosia. Yeah. So reward is, in terms of what reward is context specific, and I'm not a great believer in reward, because something that tastes like dirt is delicious if you're ravenous. Okay. And he had that concept, which to me was quite formative. And I looked at the effects of shutting down the electron transport chain to protect against reactive oxygen species, and buying that at the cost of shutting down ATP production. Now, we have a perfectly good alternative technique for ATP production, and that's glycolysis. It may be fairly inefficient, but it's extremely fast, and it can be used to fix an ATP deficit on a short-term basis. So if you take some poor person who has been eating linoleic acid to the point where they're obese, they're leaking free fatty acids all the time, they've shut down energy production, because if they don't shut down energy production, they're going to get far too many reactive oxygen species generated. If you supply them with a supply of glucose, their cells are going to be very happy. It's quite likely that their hypothalamus is going to be very happy, because it's been starving and suddenly it's been given an OXPHOS independent source of ATP. And like,
so you're in this kind of dastardly situation where you find yourself to be obese yet without energy, and you learn. This is what you're saying, isn't it? That you learn. It's learning through the experience of having energy all of a sudden when you eat glucose and produce energy through glycolysis could lead in itself to a kind of addiction cycle.
Probably light up your dopamine receptors like you. And of course, I would assume, and I've not chased the papers on this, but I would assume that fructose does it even better than glucose, because obviously, and fructose, you only have to drink a high fructose drink, and glycolysis becomes active enough that you develop lactic acidosis, not to clinic. Drinking a can of Fanta will produce lactate up at the exercise lactate threshold. It will get you that high. So clearly you have the ability to produce very large amounts in a short period of time of ATP if you're in an energy deficit. Yeah, this is again Chris Palmer's work. He's pushing. I haven't read his book. I probably ought to, but again, I try and think independently. That's how I've always worked. I can see that there's a cycle of addiction there. But how much of it is pure metabolism and how much of it is behavioral? It's an interesting question, but I can see how people can become addicted to carbohydrate, particularly if they are if they're already obese, and nobody would presumably, I presume nobody would think to blame linoleic acid for that happening, because it's 4 steps back before it triggered. But no, that has crossed my mind as to why people might be carbohydrate addicted, and how much of the complexities on top of that brain actually produces is. And again, if you're going to break a brain, how it breaks depends on, presumably there are small genetic differences that determines whether you break your brain with this ion channel, you get schizophrenia. That ion channel, you get bipolar disorder. The third ion channel, maybe you just get intractable depression.
Well, this is something that I wanted to pick up on. I was looking through Hyperlipid again, and in 2006, you did a post on Parkinson's. And you mentioned it a couple of times in other posts, and you mentioned other neurological conditions as well. But I love how George Aid talks about, there's not a sort of neurological neuron and a psychiatric neuron.
Yep.
And this study he spoke about on Parkinson's, it was in 7 people on a ketogenic diet to see if there was any difference. And the 5 people who stuck with it all had improvement on improvement on the measurement scale. And it's obviously interesting because Parkinson's is meant to be progressive and intractable.
Like type 2 diabetes.
Indeed. And so that paper has been cited by many, including recent reviews that kind of bring together emerging evidence for Alzheimer's, Parkinson's, multiple sclerosis, migraine, all of these neurological conditions. And of course, there's over 100 years of research on a ketogenic diet for epilepsy.
Yes.
So, I mean, do you make a distinction at all between neurological disease and psychiatric disease?
No, I don't think so. I think that they're all going to be metabolic related. I'd be very surprised but there will be genetic differences in terms of who gets which one, but how much of them, it looks like a very large chunk of them should be sortable by a generic approach to energy supply. And again, I haven't talked about why ketogenic diets should actually improve mitochondrial function. Because
that'd be great to talk about that. Because
well, I don't really have an answer, but you're supplying a ton of acetylcholine. Yeah, mitochondrial ketogenic diet doesn't really supply anything other than fatty acids, which fine for your astrocytes. They can convert fatty acids into ketones within your brain, or ketones directly. And you're not going to do anything with those other than use mitochondria. And given the choice, obviously there will have to be mitochondrial biogenesis, but exactly how that happens, I've not gone into that at all. And exactly the same thing will be happening in peripheral adipocytes as well. But no, I think ketogenic diet completely sidesteps the problem. In exactly the same way, it's a low insulin diet. It doesn't care about insulin resistance. You can generate enough calories from a ketogenic diet to supply what you would have been getting from insulin and glucose. And obviously, if you go ketogenic, then glucose is low, insulin is low. All you've got to do is work with fatty acids, and the system will balance itself out on fatty acids. But it takes a week of keto flu.
That's maybe if you find yourself at this point of having become obese, or functionally obese, if you like, where what you're eating, is you're leaking fat out of your fat cells, and you're not able to properly create energy anymore. When you go on a ketogenic diet, your insulin levels drop right down.
Yep.
And things start to move again, right?
Yes. And fatty acids will develop reactive oxygen species, but they'll generate them at fatty acid levels. They don't require the control system which is in place for fatty acids appears to work perfectly well, provided you're not using insulin and glucose on top of it. So again, like fasting, we're designed not to be injured by fasting, because occasionally you don't catch something for a while. But the ketogenic diet's a little bit more complicated than seeing how you might accept carbohydrate addiction as a sticking plaster on failed mitochondria. Okay, ketosis, there's a lot more goes on with that. The rule of thumb is it seems to work.
Yeah, there's so many different pathways, and any sort of ancient pathway, like superoxide generation, reactive oxygen species going back billions of years. It's hard to imagine a million years. If you think about, 2,000 years ago was kind of, Jesus and the Romans and everything. Then you go back maybe another 10,000 years, and you've got the sort of Göbekli Tepe, early civilization kind of stuff. You're already just so far into the mist of time, and you're absolutely nowhere in terms of getting back to how old these processes are, and they end up stacking on top of each other as a kind of ballet of incredible intricacy that where each chemical and step is probably doing 10 things and 10 more that we don't know about. Because it works out a way to be as good as possible in that environment.
That, and it has to be the best, otherwise we wouldn't be here. It makes me very cautious about anybody who wants to develop one drug to hit one receptor and expects to get one effect, and that isn't going to fly. It's the law of unintended consequences is unavoidable, and very powerful.
And I guess like the big question then is, if the ketogenic diet is an intervention that can sometimes be modeled as drug-like, certainly if someone is on drugs, from their doctor, then if they suddenly decide to go keto, then having access to their doctor is advisable, because you often need to deprescribe quite quickly and repeatedly. So, why does the keto diet as a rule of thumb seem to work systemically and on so many processes?
Yeah, well, and I think you have to go back, I think over the last few million years, probably go back to Miki Ben Dor's work and where human beings came from, what they lived on, and what we're currently best adapted to. And why? Well, people who couldn't cope on a ketogenic diet have not left offspring today. Yeah, there's been 10,000 years maybe of selection away from coping with the ketogenic diet, but even in agricultural times, then babies have been ketogenic for as long until they're weaned, basically. So I tend to subscribe to the concept that it's the evolutionary appropriate diet. It's the fallback, or the fallback is quite the right word. It's the default if you are having problems with a more modern diet. And why that's a very interesting question. The whole concept that, yes, the numbers that Chris Palmer throws around are that a third of people will go into complete remission for bipolar or schizophrenia. A third of them will get a marked improvement but won't get complete remission, and a third it does bugger all for. Yeah, and you go back to people like Amber O'Hearn, who were keto for decades, probably. I'm not quite sure how long Amber was before she went carnivore. With
10 years, give or take, I'm sure. Maybe 15.
Yeah, thereabouts. And then she went complete carnivore, got rid of all plants, and went into bipolar remission within 7 days. So that's why. Okay, what are the plants doing that should facilitate the maintenance of an energy deficit problem within the brain? That's a really, really interesting question to answer. And then the next move, obviously, is going carnivore is one step beyond the pale socially. Going keto carnivore, keto, I was going to say keto AF, but you know what I mean.
Keto animal foods, but
yeah, whatever. Something like that, maybe. Which I suspect is your third step. How many people who have not got the insight and the background knowledge would accept that that is a necessity to control certain processes within the body? But it appears to be the case. And I think trying to pick at those things, I think some people ultimately just have to accept that either they're going to have to do that, or they're going to have to live with their metabolic problem. But why should that flick occur from deep ketosis, and it can be very, very deep ketosis, to carnivore mild ketosis or carnivore deep ketosis, produce such a change? And within 7 days, this I have no handle on at all. I do not understand that. I would love to understand that. That's a fascinating problem to try and get to the bottom of. But again, in terms of helping people, from my point of view as a fairly ketogenic carnivore, the concept of going back to eating ketosis as a mixed diet ketosis person, it's dead easy. It's socially completely acceptable. You know, you go around to the in-laws' house, it's roast beef. You just have the roast beef, put some butter on it, maybe eat few bits of vegetable as well if you're feeling like making people happy. She's probably made keto ice cream or something like that. So it's not difficult. You're not a social outcast, particularly if you're dairy free. Sitting down and eating all of the roast beef joint and leaving none for anybody else, that ain't gonna fly. So keto is easy when you've played with it for as long as I have. Carnivore is slightly more difficult, and keto carnivore is just you're out on a limb socially, and I can understand completely that everybody can do it. But then it depends, A, on what your medical pressures are that want to make you improve, and B, how convinced you might or might not be by the arguments. And I find it very difficult not having an argument for the elimination of plants. I know they're toxins. I accept that the primary site of injury for most plant toxins is the gut. That's where you put the plants. That's where they're going to provide the greatest concentration of toxins. But why should that affect metabolic syndrome or neurological energy metabolism the way it seems to? That's a really, really interesting question. And again, I've got no idea. They're the great things. Not knowing the answer is a source of fascination to me.
Yeah, and it makes me think about the sort of the two legs striding forward in physics. They talk about, of theory and experiment, and how we have places in society for the likes of Sean Carroll, a theoretical physicist of some fame, who talks about things like string theory, which are basically untestable as far as we can tell, but have some credence because the maths is cogent. So you say that's probably how the universe is. But so what, in a sense? It's interesting, but what actually happens? And similarly with, keto versus carnivore for mental health, you kind of have to look at what happens, regardless of the theory around it.
Because pragmatics.
Pragmatics. What happens in your life is really what most people care about their lives. This an endless theorizing about what someone could do or could feel like if this, that, and the other thing. You really have to try it, right, and embody it, and dip your toe in. And this is something that I work with clients on, and a lot of people do come to the idea that keto can be helpful for mental health, and some of them start with the idea, what's a carb? You're starting from scratch, and they can just about get their head around the idea that you just cut out the starch and the sugar, the bread, the pasta, and have some protein and veggies that you like, and add fat that you like, and that's a kind of established ground that most people can deal with. But like you say, there's 2/3 of people who try keto for mental health or neurological health where there's some effect but not as much as they would like. And like you say, selling carnivore is a problem for multiple reasons. So it actually, it's going to be very hard to ever work out whether that is the missing piece. But another point is, I did the October experiment in October where I just ate McDonald's triple burgers without the bun, though I did have the condiments. And then I supplemented that with egg yolks cooked in beef dripping. That was all I ate for the whole month. I felt absolutely brilliant, mentally very sharp, loads of energy, lost 6 kilos that I didn't know I had to lose. I looked thin. It was quite remarkable, I think. And then in February, which we're just coming to the end to, I embarked on grass-federy. So I was having grass-fed beef and lamb. A lot of it was from an organic pasture for life farm. I included some organ meat, and within about 2 weeks I felt mentally sluggish and I couldn't grasp my words, and I compared it to October, decided that maybe the egg yolks was the difference, supplemented with choline, immediately got my word recall back, and decided to abandon the experiment. And, you know, that was carnivore, but it didn't work for me. And so this sort of 2/3 of people who try keto, there's several levers you can pull. There's the calorie source, there's the micronutrition, and there's the balance between fat and protein, I guess. Plus this cultural element where, what do you do when you go around to the in-laws?
Yes. Yes, quite. There's clearly more to it, and that's fine. Life is always going to have more to it. It would be boring if we had all the answers. And but no, I can see on a practicality basis that all meat is socially problematic. I've always viewed egg yolks as a multivitamin. How intrinsic that would be to human beings. Again, if you're in a temperate climate, you're not going to have egg yolks all the year round. That's for sure. So why and what on an individual basis makes people cope or not cope? I don't know. There's too much to go. There's too far to go yet. And practically, I think if you were going to recommend an all carnivore diet, I think a bit of choline wouldn't do any harm. But that's your N=1. What other N=1s people have of their own individual variation? I think it's way too early to know these things yet. And you've been, in some ways, you could say you've been lucky. You had a control setup, and an intervention that you could. And it must have been quite shocking that the whole grass-fed thing wasn't enough.
It was actually. There's decent evidence that the beef and lamb I was eating was more nutritious than the already pretty good McDonald's beef, which is just 100% British beef, and we're good at growing beef in the UK. But yeah, it was a bit of a shock. You hear so many success stories of people just eating muscle meat and not even the organs, and I think that is the key, is this N=1 experimentation. And really, I think that's another X factor here, is that people finding the confidence to do that and be different is something that's not in some people's characters.
Yes, yeah. Ultimately, and again, there is a continuous selection pressure in terms of humans are in the process of adapting to a modern diet. Yeah. So ultimately, if you did it for long enough, there would be a group there'll be a population that were the ones who coped best with donuts fried in sunflower oil, and they will be the people eventually, if we don't change matters, they will be the people who survive. But the process of acting on of natural selection is based on reproductive failure. And actually, I don't know how many young people of childbearing age, but an awful lot of the ones I work with have had to have assisted reproduction problems. So we're at the moment, medicine is fighting against evolution. So these people, lovely though they are, lovely though their children are, on an evolutionary basis, these are not the people that are well adapted to a modern diet. But again, the other concept from Nick Lane is that evolution can be very, very fast indeed. People think of it in terms of being slow because it happens over billions of years, and then you get the explosion of hard animals. Anyway, but given a sufficient selection pressure, evolution can happen very rapidly. So that in the fossil record, it appears to be instantaneous and explosive. So evolution is not always slow. If the conditions are stable for a very long time, then evolution is obviously going to be very, very slow. But at the moment, the environmental conditions are changing very rapidly. And we would probably be evolving very rapidly, except for medicine. Medicine puts sticking plasters on the damage that is used to eliminate people who are not adapted to the modern circumstance. It applies. Again, we go back to Miki Ben Dor and starch adaptation in humans, and the fact that there's a bell curve. There's a 5% of people really are horribly badly adapted to starch consumption. 5% at the other end of the bell curve are absolutely utterly fine on it, and everyone else is distributed between the middle, and you might be on the upward slope or the downward slope, so you may have a lot of pathology or not very much pathology, depending on all sorts of things, like even like your background genetics. Like if you carry the HLA-B27 white blood cell marker, you're going to do badly on starch, you can cope perfectly well as HLA-B27 positive if you don't eat starch. So it's common in the circumpolar regions. If you from tropical equatorial regions, HLA-B27 is very rare, because you're not going to get on. You can anybody who carried the HLA-B27 gene developed ankylosing spondylitis or anterior uveitis in the eye, and these are not long-term survival problems for a hunter-gatherer. So there's a distribution in terms of what was and wasn't available food-wise, which determines because people have evolved, not because evolution has a plan to make you cope all right with living on walrus meat up north, but because people who couldn't tolerate eating starches died out in the equatorial regions and survived up north. And there's thought to be an infectious disease advantage to HLA-B27, which offsets the problems it causes on an autoimmune basis. So
this is the thing, that these bell curves and distributions of an individual's response to various environments. It's not just a single isolated bell curve for starch, like you say. It intersects with some other thing, like we were talking about earlier, how evolution's had so long to try stuff that you get considerable multi-use chemicals and genes and so on, and then you start introducing epigenetics, where the epigenetic response to the environment can be extremely powerful. Like my favorite book on that is the Nessa Carey one, the Epigenetic Revolution, where she uses the analogy that your genes can be the same, like the script for the Romeo and Juliet, the Zeffirelli's Romeo and Juliet versus the Baz Luhrmann one. The script's the same, but the expression of it is wildly different.
And again, that will come down to acetylation and glutathione and all of the in terms of what controls these things. That will be again, it's a huge subject. It's interesting, but for me at the moment it's too big. It's too big. I'm still stuck down at my basic. This is the bacterium living inside an archaeon, and that's what we call a mammalian cell. Do you see yourself going deeper and deeper, or hanging up your spurs at some point, like the blogger you were talking about? I have no idea. I just do not know. And I took 6 months off during the summer this year. I'd stopped blogging. If I've got something to, partly I stopped blogging because I could not fit medium chain triglycerides into the protons hypothesis, where it's the ratio of NADH and FADH2. It just doesn't fit. The medium chain triglycerides have to be being processed differently, which annoyed me until I accepted that, well, actually, at the other end of the scale, DHA is processed differently as well. So, the body has taken evolution down to C16, or metabolic control down to C16 to C18 carbons. Anyway, that's palmitic versus linoleic, right? Sorry, the bug in the ointment's linoleic, yes, yeah. But that really got to me, because that and it took a while for the penny to drop that it's not the end of the world, that not everything fits in as I want them to. They can't be right about everything. But I don't have a plan. I just try and understand stuff. And some things interest me. Some things absolutely make sense, which is great, box, tick. Some things just don't make sense at all. And again, they're the interesting ones, but they're interesting if you can make progress. If you end up with, well, this doesn't make sense, but I can't understand why it doesn't make sense, that's just frustrating. But I suppose I started blogging, 2001, 2002, something. It was quite a while ago. And I only really blog about stuff that I'm interested in. People have made suggestions over the years. Oh, what about this? What about that? Well, unless my brain grabs it and goes, oh, yeah, that's exciting, I've got no chance. I passed a number of exams over the years, and I am appalling at passing exams. There are people who have a revision plan and summarize the revision plan and then take it down into smaller bites and smaller bites and smaller bites until they end up with the whole of a given subject in 2 sides of A4. And they walk into their exam having memorized the answers to every question that could possibly be asked. And I class that as cheating. Learning the answers, even without knowing the questions. Me, I kind of try and understand a subject, and I hope that I understand enough to wing it through the exams. So I never got A stars in any of my exams throughout the whole of university. I worked on the basis that if I understand it, I should be able to answer most questions that they come up with. And I think I got kind of middle of the road grades probably all the way, apart from that time in pathology. That was embarrassing. We started my 4th year pathology viva, and we were talking about chicks and yolk sac peritonitis, and I was having a blind panic because I wasn't very good at pathology. And we were talking about yolk sac peritonitis and whether it was an internal problem or an external problem. And it's internal because that's where the yolk sac ends up. I couldn't come up with an answer for this, whether it was internal or external. And my pathology examiner turned to me and said, "Mr. Dobromylskyj, have you ever seen a day-old chick?" Because obviously they haven't got a yolk sac hanging out the outside. Oh my God, things got better after that. They did pass me. I didn't have to do resits on pathology, but God knows how. So I would basically learn general. I'd have to understand the subject, and then I could usually wing it on coming up with a reasonable exam answer. It didn't always work, but that's the way I've worked, and I've never been terribly good at sitting there with a revision plan. So if I wanted to come up with the theory of everything, then I couldn't, just because I don't have that global perspective. I have things that interest me, they catch me, and I follow on. Well, if that's true, then that should be true, and that should be true. And then I go and look for confirmation bias. I find the studies that fit in. And while I'm finding the studies that fit in, I find the ones that don't fit in as well. And they're really exciting. So I hope that I have some degree of control over my own confirmation biases, but I'm very, very aware that they're very, very real. Yeah. But that's just how I work. I don't have a plan. I don't have, when I started the protons thread, I had no idea where it was going to go. I shouldn't have called it protons either. It was called protons again through, it's Nick Lane's fault, and he was talking about the proton gradient in hydrothermal vents and how this drives metabolism. And ultimately, I ended up not so much in the protons, but in the control of the electron flow that generates the proton gradient and what happens to that electron flow. It's again a bit like Brad, electrons in, electrons out. They go in where they should and they pop out not where they shouldn't, because 4 and a bit billion years is a very long time. Nothing happens accidentally. They do the best they can with the circumstances that they're given, which we give them. So that's where I came from. and I don't have a plan. And if I lost interest in various aspects of blogging, I would just stop blogging. But if something comes up that's interesting, I'm perfectly willing to spend a couple of hours and stick a post out there. A couple of hours to write the post, 2 days to try and find all the typos, grammatical errors, and logical inconsistencies that I've typed in. But that's what I do, and it's these are things that interest me. If they didn't interest me, I probably wouldn't do it. So yeah, that's where protons came. It's the wrong name. It should really be electrons or superoxide, because that's ended up more core to me.
Yeah, for people confused about the idea of protons and thinking, well, what's this got to do with radioactivity or nuclei or anything? It's the hydrogen atom with the electron taken out, right?
Yeah,
that's a proton.
Yeah.
Yeah. I mean, it's been amazing seeing the reaction over the years to the Hyperlipid blog, and I was just so grateful to find it as someone who studied physics and to think about it from first principles. And I think I'm a bit like you. I struggle to learn things by rote. What I really like to do is to learn learn how to think, not what to think.
Yes.
And then you can always access any information you need. So it's not about learning factoids. It's about trying to re-examine your biases and your way of understanding the world so that it's fairly reliable.
Yes. That's what I like about the protons concept, is that it has quite good explanatory power. It falls down because I hadn't realized how uncoupling was related to how important uncoupling was to fatty acid, to polyunsaturated fatty acids. So there were weird things like if you gave a rodent model 70% sunflower oil as its calories, it lost weight, but it's almost all linoleic acid. And so that, again, there are it's not complete. The protons concept is not in any way complete, and I keep finding bits that I'll add on to it, but nothing that makes me go, no, it's junk, it's completely wrong. So far makes sense, and it has reasonably stood the test of time, but it's messy because I don't have all of the pieces of the jigsaw. But if this piece fits there and this piece fits there, and I know this piece has got to be that shape, I'm perfectly willing to wait until the odd shape piece comes through because I know what I'm looking for. Yeah.
And like you say, you end up with interesting adjuncts around like MCTs and what Brad does around saying, drink your pu'er tea, and maybe there's a few other hacks which allow you to work around or show up notable exceptions or avoid foods that might cause problems, or other lifestyle behaviors that might cause problems.
Yes, quite. Yeah.
Even this amazing feedback to do with light and metabolism.
Yes, yeah, there's loads of stuff there. Yeah, and again, how do you fit that in? It's again, one always has ideas, but it doesn't go back to bacteria and hydrothermal vents. And again, I've just been driven back in that direction, possibly because I'm lazy. I don't have to, because if you take it all back to the most basic level, it's very basic. And then the explosion that goes on from there until today, and not only getting to be eukaryotes, but getting to be multicellular organisms, and then multicellular organisms with circulations and control systems. It gets beyond that. It was trying to get something simple. That's what I always look at.
And then control systems that include a lateralized brain who kind of has its intuitions hijacked by a kind of prefrontal cortex that thinks it knows everything, and becomes the kind of main part of parts of academia saying that we should be eating vegetable oils because we've just discovered that that's actually better for our hearts than eating foods that we've been eating for 3 million years, etc.
Yeah, the stuff that comes through that's just, I can't remember what the last one was, but you occasionally realise that oh, that was right. I think there was a Twitter thing that oxygen consumption by cell preparations does not automatically signify oxidative phosphorylation. I think Seafried has just published a preprint on that basis, came out. I saw the link this morning. And you realize that people have been using oxygen consumption measurement from cell culture as a marker for oxidative phosphorylation. That's it's always been the case, and it's wrong.
Wow.
Yeah, cell surface oxygen consumption is the ability on the cell surface to convert NADH back to NAD plus without using the mitochondria. So that's on the old cell surface, the archaeal cell surface. You can do that, which allows you to run glycolysis down to phosphoenol pyruvate and then divert it through to anabolism for your cancer cell to make new substrate, new substance. But if you divert glycolytic metabolites like phosphoenol pyruvate, it can't carry on down to lactate to balance the NAD plus NADH ratio. So you've got to get, if you're not using mitochondria to convert NADH back to NAD plus, you've got to do it some other way, and cell surface oxygen consumption does that. You're never going to pick that up in a cell surface in a cell oxygen consumption analysis. So, and that's been done ever since people could measure oxygen consumption of cell cultures, which goes back a long way nowadays, and it's been wrong. Yeah, so you can't stand there and say, oh, Warburg says that cancer cells don't do oxidative phosphorylation. Well, some of them do and some of them don't, but they all consume oxygen, but it's not necessarily being consumed by the mitochondria. It could just be another way of regenerating NAD plus to allow glycolysis to continue. So basically it's a way of sidestepping using mitochondria at all, really. So that was that. And the other one is metformin. People use 1,000 microgram or 1,000 micromole of metformin in all of their experiments, and that's going to kill you. But if they don't use 1,000 micromolar, it doesn't work. So all this concept that metformin is a complex 1 inhibitor, yeah, fine. If you're dead in the morgue, that's perfectly okay. And this is done again and again and again. But you can extract useful information when they dose a rodent model with metformin. Now, you know that one's not got 1,000 micrograms, 1,000 micromole metformin, because the rabbit or the rat be dead. So those bits of whatever experiments they run are completely usable and contain useful information. The minute they start on cell culture and go, well, we think that metformin's concentrated in the mitochondria because blah, blah, blah. No, no, it's not. And that's just junk. And that's been done for decades, again, as long as there's been research on metformin. And we make mistakes.
Yeah. We have to try lots of things to get
some of the mistakes.
Good results.
Yeah. Some of the mistakes are very persistent. And
I'm thinking about the Alzheimer's research that's been to be essentially fraudulent. That billions of pounds worth of research has been done on that single shaky peg.
Yes, absolutely. No, we're humans. We make cock-ups completely, and we're very loath to admit that we've made cock-ups. That's what, somebody got taken in to build the pyramids. This goes back a long way. What they expected to gain from building the pyramids is beyond me, but apparently it wasn't done by slaves. It was done by people who went in there willingly in the agricultural quiet time of year, and were convinced by their then ruling elites that building this big pyramid was a good plan.
Tilting at windmills.
Yes. Yeah, we do that.
Well, I very much appreciate all of the tilting at windmills you've done, and you know, the Hyperlipid blog was like you know, one of the sites that I read back to front a few times, and over the years. But even I sort of devoured it, I think, before starting low carb, because culturally 7 years ago, keto was kind of a weird one. Atkins had been and gone, and was probably more of an American phenomenon. And you're just so concerned that something bad's going to happen if you deviate from the norm in any way. And you know, you helped give me the confidence, and I know that you've helped give other people the confidence, and you've really illuminated a lot of these kinds of concepts. So I appreciate it.
You're very welcome. I'm glad. That's nice. You know, I just look at stuff and decide whether it makes sense or not. And sometimes it does, and sometimes it doesn't. And I'm willing to comment on that. That's how it goes. That's how the blog works. And
I mean, I'll link to Hyperlipid if people haven't been before, but it's high-fat-nutrition.blogspot.com. Is there anywhere else that you would want people to find you?
No, there's only Hyperlipid. Well, I suppose I'm on Twitter, but I don't. Facebook I use for family photos, pretty plants, pretty flowers, odd picture of the river if it's a nice day, that kind of thing. I don't do anything technical on Facebook at all. Twitter is more. I think my Twitter handle is Peter in Norfolk, and I will make snide comments about safe and effective things and things like that on there, and occasional technical posts. I don't do very much socializing there. I don't really do the kind of pretty flowers, things like watching the snowdrops come up and watching the winter aconites come out. To me, I love that time of year. It's I used to get seasonal affective disorder. But since carnivore, the winter is just another time of year. But aesthetically, spring and summer to me are better than winter. The winter's got its upsides. No, Peter in Norfolk, or at Peter in Norfolk with the. You better put it in the links. I think.
Oh, well, yeah, I think there's an underscore in there somewhere.
It's badly spelt. I had no idea how you set up a Twitter handle. I still don't really know very much how you set up a Twitter handle. I don't know how you do multiple ones or anything like that at all. I've just got the simple, well, I can comment here, and I do that if I feel I have something useful to contribute, or if I'm particularly fed up with our powers that be.
Well, I really appreciate you coming on the podcast as well. Thanks again, and hopefully it's another well-received episode.
Oh, thank you very much. It's been great talking, Ali. Cheers.
Thanks for listening and watching, everyone. Please hit subscribe where you're watching or listening so you don't miss out on future episodes. And please leave a review. It takes 30 seconds and really helps get these exciting messages out there. And if you or anyone you know could benefit from a mental health tune-up, head over to metpsy.com. That's metpsy.com, where myself and psychiatrist Dr. Rachel Brown coach people to better mental health. Thanks.